Good evening. I'd like to welcome you all to this online discourse, which is supported by the law firm of Mason, Hayes and Curran. For those of you who are not familiar with Academy discourses, these are the oldest and most renowned series of talks in Ireland. The first discourses were presented in 1786. Historically, the Academy discourses were the occasion reserved for distinguished scientists, distinguished academics to first reveal their work in public. The Academy has 6 discourses a year, and in the past few years, we've had Nobel Prize winners, internationally distinguished political figures, senior European public servants, internationally eminent scholars, and they've delivered a broad range of discourses, most of which are available on the Academy website. I'm Mary Canning, I'm President of the Royal Irish Academy. Present with us tonight also are Professor Pat Shannon, who is our Secretary, Professor Stephen Gardner, who is the Treasurer, and Dr. Tony Gaynor, who's our Executive Director. I have a little bit of Academy business which I'm obliged to execute before we start, and that is the minutes of the last discourse, which was with Fintan O'Toole and William Crawley in conversation about Northern Ireland after Brexit. This took place on the 11th of January 2020, and the minutes were posted online. As no member has informed us that they have any issues with these minutes, I shall take them as approved, and I shall sign the Academy Book of Minutes whenever circumstances permit me to do so. Our speaker tonight is John O'Keefe, who is an honorary member of the Academy. We are delighted to welcome him. He is Professor of Cognitive Neuroscience at the Sainsbury Wellcome Centre and the Department of Cell and Developmental Biology in University College University College London, and he is a Principal Research Fellow of the Wellcome Trust. He's a Fellow of the Royal Society, and he holds an honorary doctorate from University College Cork. In 2014, he was awarded both the Kavli Prize for Neurosciences and the Nobel Prize for Physiology or Medicine. Professor O'Keefe is interested in the role of the hippocampal formation in spatial and episodic memory and navigation. We are looking forward to hearing much more about his research this evening. Our respondent this evening is Shane O'Mara, who's a member of the Academy and is Professor of Experimental Brain Research, Trinity College, Dublin. Following Shane's response, we will have the opportunity for some questions from our audience, and these questions will be facilitated by Professor Catherine Godson, who is the Academy's Science Secretary. If you wish to ask a question, please use the Zoom question and answer function, and we hope to have time for our distinguished guests to answer as many of them as possible. And now, without more ado, Professor O'Keefe, I'll hand over to you with apologies that, of course, I know you don't come from UCD, so I'm sorry about that glitch. Over to you, Professor.
Did you hear all that?
You're now on.
So I was muted before, was I? Thank you very much, Dr. Canning, for that kind invitation, and thank you very much for the kind introduction, and thank you very much for the invitation. I'm very pleased and honored to be here. Let me start by sharing my screen. Okay, can you see that now? Good. Can you see that? Because there's not much sense if somebody unmute and tell me you can see this. Okay. Okay. I'm going to tell you about the brain mechanisms underlying flexible navigation, and I think the emphasis is on flexible navigation. What I'll tell you about is the history of our studies of a part of the brain called the hippocampus and how it's involved in navigation. That'll take me through the first third of my talk. I then want to share with you some of the more recent work that we've been doing in our lab, which is mostly unpublished, because I think we're beginning to actually understand, really almost for the first time, flexible navigation and how it's underpinned by the activity in this part of the brain, the hippocampus. And then towards the end, if I have enough time, I'd like to broaden out some of our ideas about how this work applies to human spatial language. Most of what I'll be talking about is work that's done on rodents, but we think it's very relevant to some of the work on human beings and particularly to human spatial language. So let me begin the first set of slides. So the problem, and as we've known it for many years now, is how does the brain enable us and animals to find our way around familiar environments? And I must say, right at the very start, one of the real advantages of studying spatial mechanisms that we think that many of the mechanisms which we will be able to uncover in our study of animals are shared by human beings. There's a lot of similarity between the way we navigate and the way animals navigate, and it's a good way of finding out how one of the basic functions of the brain is instantiated. So let me start with the problem. If someone wants to go from the forecourt of Trinity College here and wants to make their way to the Royal Irish Academy, and they ask you, how do I get there? One could actually just very simply give them what we would call a route instruction. You could tell them, well, you're facing south, and if you go to the corner of Nassau and make a left-hand turn, and then proceed down to the corner of Dawson and make a right-hand turn, you will come very soon to the entrance to the Royal Academy. On the other hand, we could fish out a map, and if we're fortunate enough to have this map of Dublin from 1834, and show them a picture of where they are on the map, where they're located outside of Trinity College, and where they want to go, and let them figure out how to get there on their own. And of course, from that map, they could actually navigate in exactly the same way, following the same route that we might have given them in the first place. On the other hand, since having a map provides you with the information about many different possible routes, they might take a different route. They might, for example, carry on down Grafton and turn into St. Anne's, and then find themselves at the Royal Irish Academy, or they might carry on down Grafton and to St. Stephen's Green, and then come back up going north on Dawson. So one of the things the map gives you is the ability to navigate flexibly. You're not confined to just one route. And it's been very difficult over the last years to try to understand exactly how a brain system could provide this flexible navigation, what kinds of representations, spatial representations it might provide, which would now allow that kind of navigation. Now, one of the ways there, as Dr. O'Mara will tell you, there are several ways in which you can approach it. You can approach the part of the brain and its the functions that it subserves. And I'm going to try to give a little bit of an advertisement for the way in which we have approached it, and contrast it to a very different approach. So the area, this is a wiring diagram of the visual system, which was produced in the 1970s by David Van Essen. And you can see right at the very top of this wiring diagram, labeled HC, is the part of the brain we're interested in, the hippocampus. And if you then go all the way to the bottom of the wiring diagram, you find yourself at the retina and the retinal ganglion cells. And if you look at the relationship between information coming in, visual information coming in from the retina, and how many stations and how many crossings and how many different types of interactions the information has to go through until it gets to the hippocampus, then you can imagine it would take many, many years if the approach you took was to start at the visual information coming in at the eye and moving way station by way station to see how the information is transformed. And so you go, for example, from the eye through the geniculate to the visual cortex and so on and so forth. A different way of approaching it, and a general way of a different way in general approaching the study of different structures in the brain is actually to start right in the structure you're interested in, to start in the hippocampus. And when we started pursuing this course in the early 1970s, we were told that this is never going to work. The information would be too complicated. If we looked, for example, at the cells in the hippocampus, we would not really be able to understand them. On the other hand, if you think about it, the information that's found in the very early stages of the visual system, right in the retina, for example, might actually be very difficult to understand, because there what you're looking at is a system which has to cope with the physics of the environment, with photons, with all of quantum mechanics. And it might be easier, and it has, I think, proven easier to start in the middle, because most of what we think about the brain in terms of our own introspection and in terms of our ideas about how our brains and other people's brains and other people's minds work come from what we could call folk psychology. We think that there are perceptions, and we think that we have an idea of objects in the world, people and places and such. And it might be that our use of that kind of information might help us to understand the activity of areas which are much farther away from the periphery, because it's probably there that these perceptions and memories and representations are formed. And that's what we've been doing. We've been actually looking at the activity and the functions of this part of the brain, the hippocampus, which is very far from the periphery. And what I want to do in the next couple of minutes is give you some idea of what we saw when we first started looking at that part of the brain, and what kinds of information has accumulated over the ensuing, I guess, 40, 50 years now. So here's the area that we're interested in, the hippocampus, and seen on your upper right, upper left here is colored in red, is the hippocampus in a human. It's buried inside of the temporal lobes, and really quite inaccessible from the outside. This is shown on the upper right. Is in yellow is what it looks like. It looks like an upside down worm. And it was almost called the Latin name for worm, but in the end, it ended up being called the hippocampus. Now, we can study the functions of areas of the brain, such as the hippocampus, by using magnetic resonance imaging and electrical recording from the scalp. And we can also look at the changes in patients who have had the misfortune of having damage to those areas. But as I'll tell you in a moment, it's generally believed that the way in which many areas of the brain, maybe most or all of them, operate is on the basis of the activity of single cells. And if you look at a series of single cells in, for example, an area such as this, and as I'll show you in a second, what you find is that cells which are right next to each other are performing slightly different functions. And so recording from a large area, as one typically does in imaging studies in the human, is going to make it very difficult because we're looking at the activity of many, many cells. It's going to be rather difficult to figure out what the function is. So what we tend to do then is we tend to record from animal models and try to give them tasks which are actually similar to the tasks we might ask humans to do, and examine the individual cells within that structure. If we look at the rat hippocampus shown in the lower left-hand side here, it's in a slightly different part of the brain from the human, but it has exactly the same anatomical structure, and there are lots of reasons to believe that it has many of the same functions as you would find in the human. There are obviously going to be changes in those functions. Human beings are not little rats, but we think that if we understand how the system works in a rat, then we'll be at least part of the way towards understanding how it works in humans. If we take this little section here through the rat hippocampus and we look at it, what we see is it's anatomically beautiful structure, highly organized with all of the cell bodies, which we call pyramidal cells, sitting all right next to each other in this single layer here. And all of their receiving processes, their dendrites, which receive information from other parts of the brain, and in many cases from other parts of the hippocampus, are all running at parallel to each other, as you can see here. So it has a beautiful structure, and it's favored of the anatomist, and it makes it very much easier than in many other parts of the brain to actually understand what the cells are doing, because we can place our electrodes right into this area where the cells are, and get some idea of what they're doing. So a typical experiment that is a modern equivalent of what Jonathan Ostrowski and I did many, many years ago. It goes as follows. You under deep anesthesia, you place very tiny wires, coated wires, electrodes, or in this case, what we call tetrodes, bundles of electrodes, into one of these cell areas in the hippocampus. You allow the animal to recover over several days and weeks. And then you ask it to go about its business, finding food, eating the food, interacting with other animals, and so forth, at the same time as you are actually monitoring the activity of these cells in the hippocampus itself. So typically we would put the animal into a box of a particular shape, in this case, a circle. We would have little lights on the animal's head so we could watch where it is with a television camera, and we could record the activity of these cells so that we can know where the animal was, what it's doing, and particularly, for example, where it was when the cells became active. And I'll show you a little video on the lower left-hand side of the screen here, which gives a very good impression of, it's a cartoon, but it gives a very good impression of what the cells do in such an experiment. And what you'll see is the animal running around in the circular arena. You're looking at the top-down view of the arena. And the animal is interested in finding food, which is randomly thrown into different parts of the box so that it can't predict where it is. So it randomly forages around the box looking for little bits of food. And what you see is here's the animal running around. And for most of the time, these cells are inactive. They don't say anything. They don't signal to the parts of the brain. But every once in a while, the cell, as shown by these red dots, becomes active, and it becomes active in a particular part of the environment. The cells are interested in where the animal is. They're not interested so much in what it's doing, why it's doing it. They're interested primarily in where the animal is. So as you can see, as the animal goes around this environment, the cell only becomes active in this western part of the environment. Now we can do a little bit of analysis of the data. We can take into account, for example, how often the animal has been on the right side of the box or the left side of the box, and we produce a map of what that cell is interested in, as shown on the lower right-hand side here, a heat map showing that the cell is most interested in the red area, and then it's less interested in the warmer colors. And the deep blue shows that it's not interested in at all in the other parts of the environment. Well, if we look at several of the cells at the same time, as shown in the next slide, what we see, and this is 32 cells recorded at the same time, what we see is that each of the cells has one of those nice little heat maps showing where it's active. And some of the cells are active in the top part of this now is in a square, in the top part of the square. Some are in the bottom part of the square, some are on the west side of the square, and some on the east side of the square. And when you look at that, you get the impression that wherever the animal is, there's going to be at least one cell active. Now, it turns out we're only looking at a very small number of cells. In the hippocampus, there are hundreds of thousands of cells, and we're only looking at 32 of them. So you can imagine that as the animal goes around its environment, you find lots of cells firing in each of the locations that it visited. Interesting, there doesn't seem to be any relationship between the cells in the hippocampus itself, where they're located. And you see here I've put an array of about 10 cells. And there's no relationship between they are physically located in the hippocampus and where the cell actually is active. So if you look at this cell here, for example, it's active up in the north part of the box. Another cell which is very close to it is active in the south part of the box, and this third cell is fired in the west part, the east part of the box. So there's no particular mapping of the environment onto the surface of the hippocampus, but yet when you take the activity of a lot of the cells together, then they actually map a particular location. Well, if you look at this, you might ask yourself, well, what is going on here? And when we first saw this, we thought, well, maybe this is providing something like a map of the animal's local environment, something very similar in a way to the map of central Dublin that I showed you earlier. Now, just having a group of cells which tell you you're in one place, for example, outside of Trinity College or outside of the Royal Institution, is not really enough to give you a map, because then all you have is a collection of place representations. What you really want for a map is to not only have those place representations, but some way of knowing what the spatial relationship between those places are. So what you would like is to know that Trinity College is in a particular direction and a particular distance from the Royal Irish Academy. And the way of thinking about what those connections might look like is to think in terms of a set of vectors. So you might think of them as a set of pointed arrows which connect together, for example, these two locations, and connect together also the locations which we traversed when we were first talking about how to get from one place to another. An easy way of understanding this is to think of the locations in an environment as represented by a pattern of activity across the cells in the hippocampus. So Trinity College might be represented by this pattern of activity A. The academy would be represented by this pattern of activity B. And these patterns of activity would be connected in some way by vectors, which actually tell the system if you're going from A to C, then you should head in a particular direction for a particular distance. Now, the power of this system, if this is what's happening, and we think this is actually what's happening, is that if you want to go from A to C and you want to follow vector AC, and all of a sudden you find that you can't go that way, the system will then generate another set of vectors, which will take you, for example, from A to B and from B to C. Sorry, this should be the other way around. Okay, so when we first proposed this, we didn't know anything about what those vectors might look like, where they might be represented in the brain. And so we and many other labs set about trying to find out where they were and what they might look like. And we now know, on the basis of work some by Shane O'Mara, who will be talking after me, and labs in America and in Europe and many parts of the world now, that there are many different kinds of spatial cells. So the spatial cells that we first found in the hippocampus here, the place cells, which are shown with this heat map, and which is shown in this polar plot to not care very much about the direction in which the animal is facing, are only one of many different types of spatial cells. Very soon after we had discovered the place cells, Jim Ronk in New York showed that there were another kind of spatial cell, the head direction cell, which is found in a slightly different part of the overall hippocampal system, which you see here in the middle. And these cells don't care about where the animal is. They care about the direction in which the animal is heading. So they are happy, as you see in this heat map on the upper left hand side, to fire all over a box, such as the circle that we just, the that we just saw. But they only fire if the animal is heading in a particular direction. So you can see this one prefers the northwest direction. We also know that there are other cells recently discovered by the Mosers in Trondheim, which look as though they are they provide a whole set of place representations, which are laid out in this perfectly grid-like pattern, and which you can imagine are providing something like the metric for the cognitive map. So as an animal runs, for example, towards the northeast in this environment, this cell will go brup, and then it'd be silent, and it'd go brup, and then it'd be silent, and it'd be brup, and it'd be silent. And if the animal had a way of counting the brups, another part of the brain is looking at this cell and saying, oh, there are 3 brups and he's going in the northeast direction, then it would give it some idea of how far it had gone. And then finally, there are a lot of other cells. I won't go into them in any great detail. There's a boundary cell, which is found in our laboratory, which measures how far the animal is from a particular landmark in the environment, in this case, from the right-hand wall. And these cells typically show a streak of activity running parallel to the wall and telling the animal, for example, how far they are from the wall. There are other cells which tell the animal how far they are from objects. So we have a lot of information about the cells in this part of the brain and how they actually would represent different aspects of the environment. And there are many, many labs now, and it's an aspect of what we now call computational neuroscientists. We've been able to lure many very excellent mathematicians and physicists into the field to try to put all of this information together and come up with computational mathematical models, which will do what we think the system does, enable the animal to move around in familiar environments and find its way flexibly. But it hasn't happened yet. And so what I'm going to tell you in the next part of my talk is recent work from our own laboratory. It's unpublished, which is actually giving us some of the clues as to how the hippocampus might be doing this. In order to do that, we need to have a task in which we let the animal find its way from one part of the environment to the other, using flexible navigation. And the task we've all used, and I'm sure Dr. O'Mara has used it himself, and you'll find this task in every laboratory that's interested in spatial navigation in the world. You'll find it in drug companies who are interested in drugs which affect the hippocampus, is the water maze, or as we call it, the Morris water maze, invented by Richard Morris in the around 1980. And what Richard realized is that he could construct a very, very simple task in which we could test the animal's navigation ability in a circumstance, in an environment where the animal had no local cues to point to a particular point of interest or information. And what it is essentially is a swimming pool for rats. It's a tub of water which has milky water in the tub, and where in one location there's a submerged platform which the animal can climb onto and get out of the water. It turns out that the platform is slightly submerged below the level of the water, and therefore the animal can't see it and has to remember where it is in the environment. So if you put an animal into this tub, this water maze, this Morris water maze, the first time, what you find, and you track its path as it goes around the environment, you find that it swims fairly randomly all over the place until it finally actually bumps into the platform and climbs on it. It doesn't take it very long to understand what the task is, and by the 8th trial, it's going directly from the start to the goal. I'll show you a video of this. It's just a very short video and give you some idea of what it looks like. Here's the environment. There's lots of cues around the environment. And then here's the tub, and here's the hidden platform, and here's the view from an overhead video camera. And you can see this is a well-trained animal, and he goes directly to the platform and gets up and shakes himself off. Of course, being psychologists, the experiment is then pushed the platform down so that it's no longer available, essentially, to test whether the platform is giving the animal any information. If you now put the animal in a new start position, it swims exactly to the position in the pool where the platform should be, and swims around and around, as you can see, looking for the pool, until eventually it says to itself, oh, this is a psychological experiment. They've probably taken the pool and moved it someplace. He's a clever rat. And he goes off and looks for the pool someplace else in the water maze. So as you can see from that experiment, the animal has a fairly good idea of where the platform is on the basis of its spatial location in the environment. And it knows how to get there by a direct route from every place in the environment. If you damage the hippocampus, as we did in the early 80s, you find that animals with that damage no longer are able to make that direct route to the platform, whereas normal animals and animals with damage in the overlying cortex are very good at doing it. Now, this is the workhorse of our attempts to understand how the hippocampus works and to use a behavioral task to do so. It turns out that it's not quite good enough to do it, especially if we want to understand the activity of those place cells, because as you can see, wherever we put the animal down, he goes directly to the goal. But we don't know, for example, when he gets halfway to the goal, what he thinks about the rest of the environment. We don't know what the cell thinks about going in a different direction or turning around in this location. Does the cell know where the animal is on that trajectory? We know he kind of knows where it is because the cell will fire just here, but we don't know what he thinks about the different directions and the different strategies. So in the last couple of years, my group has been trying to perfect a maze which duplicates all of the advantages of the water maze, but has fewer of the disadvantages, and which gives us a powerful tool for actually assessing what the cells and the animal thinks at any point in which it's going from a start location to the goal location. Excuse me. This is an earlier version of the maze. And consists of, in this version is 37 platforms, all sitting on pneumatic tubes, such that we can raise and lower each one of them independently. And on the top of each platform is a hexagonal structure which the animal can rest on or move around on in a very confined space. We can raise one of these platforms and confine the animal to that location. Or we can raise several of them, 3 in this case, and allow the animal that's confined on the first platform to choose one of the other 2 platforms. And so what we ask the animal then is, if we ask it to get from wherever it's found itself on the maze to a hidden to a location, which is essentially one of these other platforms. And when it gets there, we give it a little bit of food. So we can the trials are shown in outline here. We can start the animal on the blue maze, on the blue platform here. and we can say to it, okay, if you get to the red platform, then we're going to give you some food, but not before that. But the only choices we're offering you are the yellow platform and the brown platform. And of course, in this situation, being a clever rat, he knows that the best platform to choose is the yellow one, because that makes the least angle with the direction to the goal. Of course, we can give him other choices which are not so adventitious. We can give him a choice as shown on the right-hand side here, where we give him the yellow platform and the brown platform and ask him to choose between one of those if he's sitting on the blue platform. And here the best choice is, or at least the least worst choice, to put it differently, is to take the yellow platform, because that has the least angle with the heading direction to the goal. And what they can, and so they have to choose that rather than go directly away from the goal. We've run many groups of animals on this, and we've also damaged the hippocampus on these animals to see if the hippocampus was required for them to learn this task as it is required for the water maze. And you can see here the normal learning on the left-hand side, the normal learning of a group of rats. And what you see is they learn very quickly. These are 4 trials per day, so they learn within about 20 trials. And that's really one of the nice things about spatial learning, is they understand the nature of the task and they actually know what they're supposed to do. We don't have to spend many days, weeks training them what the requirements of the task are. They know very quickly what they need to do. Damage to the hippocampus has a profound effect on this, and they essentially never learn the task even over long periods of time. So this is a task which, like the water maze, is actually sensitive to hippocampal damage. We've looked a lot at the factors which make it easier or harder for the animal to solve the task, and I won't go into most of them, but I will just point out one, which is that the angle of the best choice, if we offer him 2 choices, there's always a better choice. The angle of that platform to the heading direction to the goal is very important. And as that angle gets larger, going from less than 30 degrees to 120 degrees or more, the animals fall, their behavior falls off, as shown by this blue curve. As you'd expect, the activity of the hippocampal animals falls off, if anything, faster. Now, we want to use this task to look at those place cells to see if they can tell us how the animal needs to navigate. Can use them to navigate. So here's a little video of what a little segment of one of these trials look like. The animal is on one of these platforms. We've actually improved, this is an improved version of the maze, but essentially the situation is the same. Each platform is raised. The animal is on one of the platforms. 2 adjacent platforms are raised. He has to choose one of those. And when he chooses, then the original platform and the non-chosen platform go down, and he's confined there for a while. So we can record from the cells while he's making his way through the succession of choices from the platform he originally was on to the goal platform. So let's just look at what this behavior looks like. Here he is waiting for the platforms to come up, and you can see he's looking in lots of different directions, and then he sees the platform come up, and he has to make a decision. And one of the nice things about this task is you can see him thinking. Now, sometimes he thinks too long, and we jiggle the original platform, and that's a reminder that he has to go. Now the platforms go down, and he then rotates in place for a few seconds, waiting for the next platform to come up. And it's this rotation on this waiting platform that's very important, because that allows us to assess what he thinks about the different directions. He now makes his choice, and now the platforms go down around. And you'll see again, he looks around and he's looking in all the directions. And it's that behavior of looking in the different directions which allows us to assess what the cells are doing and what they think about the different directions to the goal and elsewhere during that period of time. And he keeps doing this, and here he's now going to stop. I'm going to stop the video, and he rears up. He's quite comfortable on the maze. He likes playing this game. Okay, so now what I want to do is take you through a few slides. I'll do this reasonably quickly and just try and draw general summaries of the sorts of things we see when we do this. This will take me another about 5 or 10 minutes. So what you're seeing on the upper left here is a single trial as an animal is making his way from the location down in the southwest part of the maze up to the north east part of the maze here where the goal is. The goal is up here. And you can see shown in this white tracing the behavior of the animal. And you can see as he goes from platform to platform, he sometimes turns around on each location. Sometimes he doesn't go all the ways around, but in general, he's making at least semicircles in most of the locations. And that's the information that we need to be able to assess what he thinks about the different directions in each of those locations. You can also see from this trial, as shown in these red dots here, where the cell fires. And interestingly enough, although when the animal is given in the original demonstrations of the place coding of the cells, the cells seem to fire in any direction that the animal was pointing in. When we give them a goal and ask them to go to it, the cells start to become directional. They start to be interested in some directions on the platform and not others. So you can see the cell doesn't fire when he's looking in, for example, the west direction, and it doesn't fire so much when he's looking in the east direction. It fires mostly in the northeast direction. If we give him another trial as shown on the right-hand side, you see a very similar pattern. He circles around and the cell is restricted to a particular firing pattern in a particular part of the maze. Of course, it's a place cell. So if we look at the place firing of it, then we see that the cell only fires in a particular part of the maze. But more interestingly, we can actually see this directionality of the firing of the cell. If we look, for example, at this middle plot here, where all of the firing of the cells are shown in red. And you can see these little scallops where every time the animal circles on a platform, the cell fires in one direction, but not in other directions. If we plot all of those directions that the cell is firing in, what we see is something shown on the lower right here, a bunch of these vectors, a bunch of arrows, which overall look like they're pointing towards a particular location on the maze. And in fact, that location is sitting right on top of the goal here. And as the animal moves around, you can see the arrows change their direction as if they're hunting this location. So this vector field, this field of arrows, is actually pointing in this particular cell towards the goal. Now, that's not always the case. It turns out that in some cases, the cell fires, the cells are pointing towards different locations. If we take all the locations that the cells are interested in one animal, and we call these convergent sinks, then we see that, for example, in rat 1, they're all over the place. These red dots say, where are those cells pointing to? Here's the goal, and you see that the cells are pointing to locations all over the environment, and some of them are outside of the environment. And that's true of all 3 animals that we've looked at so far. Now, it turns out, though, that if you say, where is the center of all these areas of interest, then it comes to a place shown by these black dots, which is quite close to, in some cases, the platform, which is the goal platform. And I won't show you this, but I can tell you that the more the animal understands about the task, the closer that black dot gets to the goal. And if we look at these vector plots from all of the cells, if we look at the population of the cells, not just one cell, but how all of the cells are cooperating with each other, and see which directions they're pointing the animal to, then we find that these vector fields are all flowing towards a point which is right on the goal, as shown by this red spot here. So wherever the animal is, this is now animal 1, the cells, the population of cells are telling them go in a particular direction. And that's true of all of these animals here. And that direction quite often is right in front of them. Some of the cells telling the animal to go off to one side or the other side, but when you look at all of them, they're telling the animal go straight towards the goal. And put your nose right in the direction of going to the goal. Okay, well, we've done quite a few experiments. I won't go into too many of them, but if we do things like change where the goal is, and these representations of the environment, these vector fields and these consinks, these preferred locations, are actually helping the animal to find its way to the goal, they should change. And that's exactly what we've seen. This is just one animal here to give you some flavor, but here are the consinks when the goal is in the southeast here. And then when we change it to the northeast, what we find is that they change, and they change very rapidly within 10 trials, 12 trials, I think. And if we just measure the distances that they are from the goal, when this is the goal, they're closer to this point than to this point. And when this is the goal, they're closer to this point than this point. They're shown by these graphs here. So it looks as though this representation really is helping the animal to find the goal. Okay, so if the animal just wants to go to the goal from a particular position, and it can go that way, then this aspect of the firing of the cells will tell it which way to go. But of course, the world isn't made like that. Certainly, the human-made world is not like that. And in the rat world that we create, it's also not always like that. So let's go back to our map of the center of Dublin. You remember we was asking the person to go from this location, someplace outside of Trinity College, to the Royal Academy. And we're giving them freedom to take whichever direction they want, or take any path they want. Now, what we're saying is that this yellow vector is created by the hippocampal activity, that the cells in the hippocampus create this yellow vector. And I've shown it again here in a very schematic form as vector AB. But of course, the animal and the human can't go directly, quite often, directly from one place to the other. It would have to climb over the buildings and so on and so forth. So it needs a mechanism which is going to get it to where it wants to go, irrespective of the fact that it can't go directly there. So it needs a mechanism which will tell it to go first along Nassau, and then down Dawson, and even if that route is blocked, as shown by this green block here, the chaps are digging up the street on a particular day, to give it a means of, and to give it the information to take alternative routes. And so we want to know is, can the system generate these alternative directions and provide information about what's the best of those alternative directions? Can it generate the vectors, which I've called AD here, going straight down Grafton, or even the vectors going in directions away from the goal? And can it also say which of these directions is the best way to go? So that's what a flexible map would enable you to do. If you look at the diagram on the right-hand side, it would say, well, A to B is the best way to go. But if you can't go from A to B, then go from A to C, and then you can go from C to B. And definitely don't go heading off in the opposite direction. And even AD is not a particular way to go. So can the system do that? And I'll tell you in the next 2 or 3 minutes exactly, that's exactly what it can do. So if we look at the activity of these cells as the animal is rotating around on the platform before it has the choice. So here it is sitting on this central platform here, and it's waiting for the other platforms to come up. It doesn't know what those platforms are going to be. But we can look at its brain activity and say, well, what does it think about the different directions? Does it have an idea that going straight ahead towards the goal is the best way to go, as shown by this large red arrow? And does it know that these other directions are less good and that some of them are still better than others? Does it form some kind of a vector representation of the sort shown on this upper left-hand side here? And the answer is yes. So this is the schematic data shown on the left, and this is the actual data when we combine all of the information from all of those 3 rats making all of their choices. And what you can see is that if the animal is offered a choice between a platform which is heading just slightly off the goal and one which is heading at 90 degrees, it can just look at how big this vector is, and which is essentially how much the cell is firing, and choose between those two in terms of that vector. And it does it for the whole 360 degrees. It's a really quite astonishing finding. And it turns out that if we plot for the 3 animals, the amount of firing, or the length of these vectors as a function of the angle away from the goal as we turn either through right clockwise turning or anti-clockwise turning, we see that it falls off very nicely, so that the animal could always tell that it's better to go in 60 degrees, either to the left or the right, than to go 140 degrees. Now, remember what we're saying here. This is a little clip of the video. And what we're saying is that the animal is making this representation and has this knowledge before it has any idea of what choice it's going to make. So this is a pure spatial representation. So this is done while the animal is sitting here on the platform waiting for the next choice. And he's looking around. And that's when we look at this and find this representation. So the brain is representing the possible choices it can make and giving them a score as to how good they would be before he knows which ones he's going to have to make. And here, of course, when the platforms come up, so that's Jake, who's run this study, and reminding me to give him the credit for running this study, because he was the one who did most of the experiments. Okay, so I've got coming down to my 45-minute time allotment, and I just wanted to step back a bit from this finding, because what we're saying is that at least within the spatial domain, the hippocampus is not only telling the animal where it is, but if it knows there's a place where it wants to go, for example, the Royal Academy, it's telling the rest of the brain how to get there directly. And if it can't go directly, what's the best second choice to make? And it's giving a whole array of possible choices and ranking them in terms of their the goodness of heading towards direction. Now, one of the problems with this whole story, and the whole story about the hippocampus as a cognitive map for many years, has been that it certainly looks, if we apply it to the human, it looks like it covers quite a bit of the story, not all of the story, because we have to include a time into the equation. We have to say when the animal was in particular locations at particular times to account for all the data, but it accounts for quite a bit of the data that's seen with, for example, damage to the right hippocampus in the human. It doesn't account for what's happening in the left hippocampus in the human, because what's happening in the left hippocampus is something to do with language. And of course, one of the nice things about studying spatial navigation and spatial location in rats is that you can imagine that the system is conserved when you go from a rat through some imaginary evolutionary cycles to the human. But you can't imagine that in the language system. But I think it's widely accepted now in cognitive psychology that the language system that you see in adult humans is parasitical on other parts of the brain and prior parts of the brain which were doing something else and which were then parasitized by the language system. So in this case, what we're suggesting is that spatial language, and particularly spatial languages is represented in the left hippocampus, is actually parasitizing the spatial functions of the right hippocampus. And so to just give you a flavor, I won't go into this in any great detail because time is running out, but to give you a flavor, we're talking about the left side of the brain here, which in general seems to be dedicated more to language functions than the right-hand side. And we're asking, how would we describe the behavior of a human that I just told you about, who wants to go from Trinity College to the Academy, in terms of spatial language? And so what we want to do is we want to find some aspect of spatial language which represents the movement from A, Trinity College, to B, the Academy, in terms of these vectors, because that's how we think the hippocampus works. And it turns out that one of the ways we can do that is to focus on the prepositions. The prepositions are a closed class set of words. They're similar in, if not identical, in all of the languages that one knows about. There are a very small number of them, about 20 to 30, depending on how you count them. And if we think about it, if we wanted to describe what this, in language, what this vector looks like, and this is a vector which goes from A to B, and we don't want to describe somebody who is taking that path, then we would say, Bridget went from Trinity to the Royal Academy. And this is a mistake. The from is the one that should have been colored, because what I'm saying is that the vector is actually described by the from and to. So to go from Trinity, this is Trinity, that's the tail of the vector, and to the RAA is the head of the vector. And you can see how you could then begin to describe many of the different aspects of spatial language and spatial movements using language. And I've written several papers on this if anybody's interested. So for example, you could say, instead of saying Bridget went from Trinity to the RAA, you could say Bridget went from Trinity to the RAA via C. I took this path going to the corner of Nassau and Dawson. And you're essentially describing the vector sum of these two vectors. And what I find interesting about this is that we know a little bit about where vectors are stored in the human brain. Sorry, the prepositions are stored, and therefore the very elementary vectors, they're stored in this area of the parietal cortex here, the inferior parietal cortex, which has very, very strong connections to the hippocampus, and they're in both directions. So if we think of these basic vectors as being generated in this part of the brain. And when you do experiments in which you ask people to tell the difference between a circle and a picture of a circle and a square and a picture of a circle and a square, where the circle has a particular spatial relationship, this circle is on top of the square, then you find this area lit up. So it's very clear that it's telling you the relationship between two objects in the environment. Then that information is sent to the hippocampus, where it's further processed, put together with other vectors, and creates what I think we would want to talk about as something like the way in which language is very good about talking about the possible things you could do. And that's, of course, what we mean when we talk about the subjunctive mood. So if we wanted to say Bridget could go from Trinity to the RAA via NASA or Grafton, then that would be represented by these fantails, these representations of the environment before the animal actually knows what action it's going to take. And then when the animal knows what action it's going to take, so for example, it will go via Nassau and not via Grafton, then that collapses into the simple representation of the two vectors. So we think we're getting at a way in which the brain can represent possibilities and predictions that the animal could make even before it knows what it will have to do under those circumstances. Okay, well, that's my talk. Let me just summarize. So the cognitive map theory has been around for a long time, and it still has a bit of mileage in it. It not only identify cells in the hippocampus and the cognitive map, therefore, not only identify the animal's position and what's in those locations. It wouldn't be very much good to know you were in a particular place if you didn't know what to expect in that place in terms of rewards, punishments, other people you might meet. And in a known environment, this provides information necessary to flexibly navigate to a goal in that environment. We're getting some idea of how it does this. It sets up these a set of these preferred locations, which we call convergence states, in an environment, known environment. And when you look at them, they come close, the average of those comes close to the goal location. And if we look at the vector fields, I remind you on the right-hand side, they point to the goal from any position in the environment. So that will provide the information that the animal needs to go directly to the goal. And when you move the goal, all of these, the consinks and the vectors all change to reorganize the representation for the new appropriate goal. So this fantail, I didn't go too far into how we measure it and how we actually extract it from the data, but this fantail looks like it's representing and evaluating the desirability of moving in any direction from a particular location. It's telling the animal, if you're given a choice between any two directions, this is the one you should choose. So it gives the rest of the brain a set of possible actions. And then when the animal knows what the choices are available, it then essentially can choose between those two on the basis of this representation. Finally, I think this has implications for human psychology. Rats can't talk, of course, but language probably parasitizes pre-existing functions such as the cognitive map to form both physical and metaphorical locations and navigation. Of course, a lot of what we talk about is getting, I'm going to try and get from some place to another, not in a physical location, but in a metaphorical location. And we think that that's also subserved by the system. So finally, I've told you some of the people who have worked with us, but I want to recognize the funders. We're funded by the Gatsby Foundation and the Wellcome Trust. And together they have given us quite a bit of money to build this new institute, for which I was the original director, but I've stepped down now to go back to the lab, the Sainsbury Wellcome Center. And the Sainsbury Wellcome Center is located at UCL, where I've been for my whole career, and it's been a very nice home to be in. So thank you very much for your attention, and I will turn you over now, or someone else will turn you over now to Shane O'Mara. I should say I talk a lot about walking and navigating, but Shane O'Mara is one of the people who does a lot of walking and navigating, and is really one of the best people to discuss this this whole topic, because he is one of the experts on this area.
So thank you all. I think John, yep, I'm going to share my screen now. Hopefully we'll be able to get rolling from there. Okay, so thank you for that wonderful talk, John. And what I'm going to try and do is step back a little from the details of the experiments that John has been talking about, and to try and contextualize a little bit how we might think about understanding the brain itself. So if we take the general question, how should we investigate brain and behavior? Well, the earliest approach, of course, has been, if you look at the figure here on the left, is gross anatomy. We look at it and we describe it, but that doesn't tell us very much about how the brain generates behavior. And here on the right, we have a variety of brains in motion, one on a bicycle thinking about moving around, another thinking about food while skipping, and two people engaged in what really is the essence of our humanity in a way, talking to each other, two brains in dialogue. So how should we investigate brain and behavior? It's not, I suppose, terribly obvious. One strategy that has been adopted, and adopted very successfully, is what we might describe as the follow the stimulus strategy. So this is the brain viewed from the outside in. So here's a famous image. This is from Descartes. What Descartes presents here, and of course it's a perfectly reasonable way to look at the brain, is a person here kneeling beside a fire. Fire is warm, and the information regarding that fire is somehow passed through nerves, up through the spinal cord and into the brain. And this is a way of thinking about how we should understand the brain. And then there are natural questions that arise as a result of that, such as how do our nerves detect a sensory stimulus and inform the brain about it? How does the brain decide what response should be made? And then how are muscles moved by the brain to produce a response? And this has been a tremendously successful way of approaching the brain. We can drop down a level and actually look using some modern neuroanatomical methods, and we can look at the nature of sensory neurons. These are the neurons that bring information into the nervous system. John mentioned the retina, for example. So here's a bipolar neuron in the retina. But equally, here is a somatosensory neuron, which is concerned with touch in the skin. And this passes information to the brain, to what are sometimes referred to as interneurons, the neurons between sensory input and motor output. And these come in a staggering variety of forms. Their morphology varies very dramatically depending on where you are in the brain. You have these beautiful cells, the pyramidal cells in the cortex. You have these rather wonderful stellate cells in the thalamus, and these amazing cells in the cerebellum, the Purkinje cells. And eventually, this must turn into some sort of behavior. So a signal must come from the brain through the spinal cord to muscle, and movement of some sort happens as a result. That movement might be articulation of words, it might be pointing, it doesn't matter, but a movement results. So you have this strategy which starts at the outside with the sensory neurons, moving through the central part of the brain, or the central nervous system, and then out through motor neurons. And you can characterize this as an overall research strategy as saying, follow the stimulus from the receptor surface into the brain and look at the successive transformations of the information that's in the outside world. And this has been a tremendously successful way of looking at the brain. So here's an example. This is the ear, where sound waves are collected by the pinna. These are localized in the auditory canal. They bounce off the eardrum. And this is converted via this wonderful set of bones, the ossicles, into movement. And here at the cochlea. And rather like the scale on a piano, the cochlea converts the sound energy, the sound waves, into electrical signals. And at the end of this processing chain, you have these amazing cilia, and these swing they contract, they change their shape, and nerve fibers emerging from the bottom of these fire action potentials and signal onward the type of sound that you've been exposed to. And if you have enough of these cilia, they can sample the whole soundscape that you've been exposed to. So here's an example. I hope you'll all be able to hear the sound here. This is a single cilium. This is an electrode, and it's sitting in a dish, and a sound is going to be played to this. These are the so-called dancing hair cells. And the point here is that this individual hair cell subsamples the soundscape. It doesn't respond to all of the components of the sound that it's exposed to, but put together enough of these and you can sample the whole of the soundscape that you've been exposed to. Now, if we think about a different system, one that has been very well investigated as well, the human visual information processing system, again, this is kind of a standard model we have now of how this might proceed, where there is information or stimulation provided by the visual field. This passes through the eyes via the lateral geniculate nucleus to the optic radiations, and then eventually here to primary visual cortex, and then onward for processing in a variety of other kinds of ways. And this view of how we should consider how the brain processes information has been very, very influential. So much so that much of cognitive psychology takes a similar processing view. So this is what's sometimes referred to as the modal model, dating from the late 1960s, of human information processing by Atkinson and Schifrin, where you have environmental input transformed in some way into sensory input. This passes through a narrow attentional bandwidth into short-term memory, and then it may be forgotten, or it may end up being passed into long-term memory. Now, this is not a current model, but it's a model that grew directly out of the kinds of thinking that I've been talking about, where we consider a processing chain where we follow the stimulus into the brain and eventually try and follow it back out of the brain again. When we look very closely at the visual system, for example, this is the kind of standard model that we've got, that it divides in two. You have an input from the optic nerve going to the lateral geniculate nucleus, to the striate cortex, and to other visual cortical areas. And this is a standard textbook presentation. But there's a problem with this, and I'll come to that problem over the next couple of slides. This way of thinking has been opposed theoretically for many years. So going back to the foundation of psychology as a scientific discipline, William James stated very bluntly that whilst part of what we perceive comes through our senses from the object before us, another part, and it may be the larger part, always comes out of our own head. And 100 or so years after, or 90 years after James made this assertion, perhaps one of the preeminent explorers of the cerebral cortex, Vernon Mountcastle, was able to claim that each of us believes himself to live directly within the world that surrounds him, to sense its objects and events precisely, and to live in real and current time. I assert that these are perceptual illusions. Sensation is an abstraction, not a replication of the real world. This brings us to the cognitive map, and in a peculiar kind of way. So John has given a description of the physiology of the cognitive map. What I want to do is talk about the behavior involved. So this is a paper from Tolman, where he introduced the phrase the cognitive map in the late 1940s. And his task was very simple. He took rats, he introduced them into a maze. They would run in through the entrance tunnel. They would run forward, make a left-hand turn, run forward, make a right-hand turn, and they would end up at a position in space where they would get a food morsel. And they will learn this task very quickly, and they will be able to do it very efficiently. Now, Tolman wondered what would happen if he cheated ever so slightly. And this was his cheat. He closed off the part of the maze that the animal was used to running in, and he provided this sunburst configuration. And the question then is, will the rat attempt to run through this part of the maze, or will it be able to figure out quickly which of the other routes it could take that would get it the food morsel? And what he found in this experiment, and remember, the animal had never made this particular set of motor movements before. So that is probably the key point. It had been rewarded for engaging in a straight run, a move to the left, a move up, a move to the right, and a circuitous route. Could it make the rapid inference that it should just simply run in and run to the right? And the answer is, of course it could. And it did so very, very quickly. They would run down the correct tube, or path very quickly to retrieve the morsel of food. And what Tolman speculated, and I think John O'Keefe's talk provides the solution to, is that the rats were inducing or inferring something like a survey map of their environment. And this conferred on them the ability to flexibly navigate that environment. And this was quite contrary to the view that had been prevalent at that point, that there was a stimulus-driven component to behavior. There presumably is, but that in the case of navigation, something else is going on, that the rat was capable of making an inference about where it is and where it wanted to go, that was independent of the previous pattern of reward that it had experienced. And why might you think this? Well, there's a reason for thinking this. The follow the stimulus inwards view runs into a problem, and it's this, that cognitive and behavioral responses are not aligned along sensory axes. Instead, they're aligned along multiple axes that are expressed through the motor system, because you have to move in order to get where it is that you want to go. But that the actual learning itself is something that is not given by previous patterns of reward or previous patterns of stimulation. Now, if we come to humans for a moment, brain imaging has given us a picture of the brain at work in a way that is unprecedented compared to what was possible to do in previous years. And the image that I'm showing you here is canonical. It's almost totemic in terms of its importance. When you engage in brain imaging, one of the key things that you do is you must have a control condition. So the person is engaged in a task of some description. This might be a memory task, it might be a visual discrimination task, or something of that sort. But in order to figure out what activations are happening in what part of the brain, you must have a contrast. And the contrast that people have used has been to get people to close their eyes and think of nothing. And it turns out that actually, when you're thinking of nothing on the instructor of the experimenter, you're actually thinking of something, and the brain becomes remarkably active, so much so that this part of the brain this kind of activity is referred to as the default mode of brain activity. That there's lots going on in the brain in terms of intrinsic activity that cannot be accounted for by a simple or even a complicated stimulus-driven view of brain activity. It's estimated from experience sampling studies that we engage in perhaps 30% of our waking life in this default mode, where we're mind wandering, we're considering our own autobiographical memories, we're considering the big picture of our lives, we're thinking about tomorrow, we're thinking about yesterday, we're thinking about what it is that we are doing today. So intrinsic activity has been visualized very effectively using brain imaging, and it really is quite a remarkable discovery that there's much more going on in the brain, even under stimulus impoverished circumstances where you're lying in a loud tube with your eyes closed. Now, what I want to do is shift gear and come back toward where Professor O'Keefe's talk was concerned. And what I want to do is ask the general question, why do we have a brain? Trees don't have brains, at least brains that we're aware of, I suppose. Plants don't have brains. So having a brain says something about the difference between animals and plants. There's a different way of expressing this, which is to ask the question, what problem does having a brain solve for you? And what I want to do is tell a small story from biology about a particular animal. This is a tunicate, a rather beautiful animal which is found in warm waters around the world, also known as the sea squirt. In its early larval stage, the sea squirt has some similarities that make it part of the same phylum that we humans are part of, phylum Chordata. It has a spinal cord and it's able to swim in the water. It has an eye spot and it has a primitive proto-brain. And this is what it looks like in cross-section. It can tell up from down by using this little staticist, which is a kind of a ball which rolls around to tell it which way is up and down. It can escape predators. It has a stomach. And as I've already mentioned, it has a kind of a primitive spinal cord, which makes it part of the same part of the animal kingdom as us. And it even has a cerebral vesicle or a kind of a proto-brain consisting of a couple of hundred brain cells. Now, at a certain point in its life, it transitions from being a larva and it sticks itself using a pedal disc, this disc, to a rock. And over a period of a week or two, it gradually absorbs its own spinal cord as a meal, de-differentiates these cells, and it also absorbs its own eye, its cerebral eye, and the staticist, which allows it to tell up from down. And it mostly consumes what it uses as a central nervous system. In other words, it absorbs its own brain as a meal. And I think there's an interesting way of thinking about why we have brains. I think brains likely evolved to solve the problem of adaptive navigation in complex 3D spaces. And by adaptive navigation, I mean things like finding food, finding mates, finding safe places of shelter, avoiding predation, and the like. And to bring that right up to the kind of the current point, here's a Google Earth view of adaptive navigation. We can use remarkable handheld devices as kind of external cognitive surfaces in a way that would be impossible, of course, for our little sea creature friend to conceive of. But even the original map makers that were present in, who constructed the map in Professor O'Keefe's talk. Thank you. I'll stop here.
Okay, so if I can thank both speakers very much for a fantastically stimulating discussion, and we have a couple, in fact we have several questions that have come in. I suppose the inevitable one that has come in, that's probably directed more towards Professor O'Keefe, is if any of the trials, I guess the question is, have you always used male rats, or do you see any difference in the sex ability around directional behavior?
Yes, thank you very much. So historically, psychologists who work with rats have tended to use primarily or exclusively male rats because it avoids some of the variation that you would get with the estrous cycle in females. There's not a very good reason for doing that. And certainly within the last 10 years or so, both we as experimenters and funding agencies have started to bring that to our attention and insist that we work on both males and females. It turns out, and I have looked at female rats and looked at their place cells and the navigation, and we don't see any difference. And that's the general finding that our field is coming up with. Now, that's not to say there aren't differences between males and females in other functions, and obviously, if you look, there are areas in the female brain which are very different from the male brain. They're called sexually dimorphic areas, and but aside from those, in terms of cognitive functions and things like that, we don't see any difference. There is a lot of anecdotals and some supposed evidence that there are differences in navigation between human males and females. If you look at the literature, what it turns out to suggest is that there isn't any difference in their ability to solve spatial navigation tasks, but what there are is a difference in strategies. So it looks as though males tend to perceive the environment as a whole and look at the shape of the environment, and might go, say, to the northeast corner or to the apex of a triangular environment. And females tend to pay much more attention to objects. And if you control for those, then you find that there's no difference, or you can actually bias your experiments one way or another if you're not careful about it.
Okay, great, thanks. One more question for you, which is, do vector fields fantail representations change across multiple trials? So basically, do the headings become more accurate with learning?
That's a very good question. Obviously, this is unpublished work, and given that I knew it would be a general audience, I tried to present a general view of it, so I left out a lot of the details. It turns out, yes, that's in fact one of the things that we're seeing. If we look at, for example, when we change the goal, and then take all of the trials that we give the animal after we've changed the goal, and then break them up, say, into the first half and the second half, what we see is that all of that structure that I talked about, the actual convergence sinks and the vector fields and the fantails all change over that time, and they move closer and closer to the goal. So we do have a very strong, very rapid learning system. We're talking about very small numbers of trials, very small numbers of experiences. And of course, this is one of the things we're interested in. I should say one of the things that's surprising to us is it looks as though the representation is at the population level. There are many people working on the nervous system who say, why do people look at single cells? Because it's the activity of large populations of cells, which is in fact what's doing the computation. And I've kind of been skeptical about that for a time, but this really does look like that. So if we look at the cells which are involved in one goal, and then we look at the cells which are actually signaling that the goal has changed, they quite often, most of them are actually different cells. So one group of cells stops firing and the next group of cells takes up, as it were, the baton and says, no, this is where you have to go. So just looking at individual cells, as we have been, for technical reasons, forced to do for most of our career, is going to miss a lot of the interesting activity. And the great advance in neuroscience, as I'm sure Shane will confirm, is we now have ways of looking at hundreds and thousands of cells at the same time in freely moving animals. So we can look at the population, and it's starting to throw up very, very interesting things that we didn't know before.
And is there much plasticity between the different cell types, or are they committed to being, you know, place cells or head direction, or the grid cells that you described in the first instance?
Yeah, that's a good question. And so obviously one of the things we're always worried about is that we're only seeing the elephant's tail and we're not seeing the whole picture. We're pretty sure from experiments we and other people have done is that if you look in the hippocampus itself, that if you give the cells enough opportunities to be place cells, take the animal and move it from one box to another box to another box, then they all turn out to be place cells. Only a subsection of them are these directionally oriented cell consync cells, as we call them, about 30% in our hands. But again, I think, you know, it's early days, and what we might find is that different cells are active in different environments.
And I guess if we're to extrapolate from the level of stimulation that we're getting or not getting in the pandemic in like young kids, we could imagine that there's going to be a difference in brain activity in that regard. Maybe Shane might comment on that.
I don't know is the honest answer. I can give a slightly maybe unexpected answer, which is that we know that kids who are driven to school versus kids who walk to school, the kids who are driven typically don't know where they are when you ask them to draw maps of the route that they take. Whereas the kids who walk have engaged in this latent learning that allows them to pick up information. And this is irrespective of whether you're male or female. If you're a kid sitting in the back of the car, your knowledge of your environment isn't as good as it would be if you were a kid who had to walk.
Actually, can I just ask Professor O'Keefe, there's a question here about the honeycomb maze, and it's making a distinction that in this case, there weren't any borders, there weren't any walls. And the questioner was wondering if it'd make any difference if the cells have walls or borders around the maze, like I guess they have to have in the water maze.
Yeah, yes, yeah, quite. It does make some difference. We don't think it's going to be a fundamental difference. What you find, surprisingly, there hasn't been very much work on it, but what you tend to find is that some of the points of interest are located on the outside of the maze, on the other side of the wall. So they, but it's a very good question, and clearly the walls are a very important landmark. And we know this, for example, with the grid cells. If we change the shape of the box, say from a rectangle to a trapezoid, that has a big effect on the firing of those cells and of the place cells. So walls are very important, and our next experiment will probably be to do the same experiment with walls.
Okay, great. Well, the question suggested that. Okay, so we're at 29 minutes past 8. So I'd just like to call proceedings to an end, and thank very much, to give thanks very much to John and to Shane and to everybody for their questions. And I'm sure you'll agree it was a most enjoyable and interesting discourse. And I'm certainly going to wonder about how much variability there is in basal brain activity, which is a question I might pick up with Shane at another time. Anyway, I'd like to thank our sponsors, Mason, Hayes and Current, for their generous support of the programme. And just to remind you that the next Academy Discourse will be a conversation style event on AI through the looking glass, discussing AI and ethics with artificial intelligence experts, Dame Wendy Hall and Barry O'Sullivan, MRIA. And that will take place online on Thursday, the 4th of March at 7 o'clock. And further information and booking details are available on the Academy website. So I'd like to thank the Academy staff for their support with the event. And of course, thanks again to the speakers and to the audience. Okay, good night. Thank you.
Good night.
Good night.